Welcome to Dark Water Blues, Estuarine Community Response to an Ecosystem Level Hard Clam Manipulation. My two co-authors is Dr. Chris Gobler, who's made his career out of looking at harmful algal blooms, and Mike Doall, who is the program manager for the Shinnecock Bay Restoration Project. This work was done in Long Island, New York, in one of the Long Island South Shore estuaries, Shinnecock Bay. In 2010, Chris Gobler and I began doing a series of experiments in Shinnecock Bay, and at that time, all of Western Shinnecock Bay was closed to shellfishing due to biotoxin. This is an image of the Oreococcus blooms shading out the light in Western Shinnecock Bay. When we went further and further away from the inlet, we saw a greater and greater increase of chlorophyll. But not only did the chlorophyll increase, but the proportion of that chlorophyll that was composed by small particles also increased. At the same time, as we moved away from the inlet into Western Shinnecock, there was a decrease in eelgrass productivity until they couldn't get enough carbon to survive. We knew what the problem was, that we had a nitrogen input, and that nitrogen into our Western Shinnecock was primarily because of these aging septic systems. We have no sewage treatment out there. And so 61% of the nitrogen entering the watershed was coming from the groundwater because of these aging septic systems. And that had, changed a system that in the mid-70s was characterized by high hard clam populations, low biomass in the water column, high light in eelgrass, to a system that now was predominantly water column productivity and insufficient light reaching the bottom in Western Shinnecock. So we wanted to know, how can we drive this back? And we started to explore the use of hard clam restoration as a means for ecosystem manipulation. These two areas in Tiana Bay and Weesuck Creek, we, with the aid of the town trustees, created a no-take place for fishing, and we started to begin putting hard clam spawner sanctuaries in these areas. Since 2012, we've put over 3,400,000 adult clams into these spawner sanctuaries. You can see here deploying those in the field. These are a picture of where the 64 spawner sanctuaries have landed in Weesuck and Tiana Bay. The target densities were somewhere between 25 and 35 hard clams per square meter. So each of these received roughly 50,000 hard clams. We've monitored a subset of those sanctuaries since the inception of the project, trying to make sure that our survivalship was high. We also created a larval transport particle model, which showed that the larvae produced in those spawner sanctuaries would have an eastward and westward transport. We've been doing suction sampling in Eastern and Western Shinnecock. And as you can see here, comparing 2015 in Eastern Shinnecock to 2020, we have a lot more juvenile hard clams found in those. And this graph shows the light blue color is Western Shinnecock and the dark blue is Eastern Shinnecock. And you can see that there's been this very large, significant increase in juvenile hard clams in our sampling in Eastern Shinnecock. And when you compare the harvest collection in Eastern and Western Shinnecock, what you see is Western Shinnecock is primarily composed of chowder clams, where in Eastern Shinnecock, when you see the differences in kind of the numbers, but also in most recently that they're composed primarily from the smaller clam sizes. So when we look at the hard clam harvest in Eastern and Western Shinnecock, here's the restoration initiation. You can see that there's been this significant increase in harvest from Eastern Shinnecock. When we look at the size structure of those hard clams being landed, we see that there's this disproportionate size of little necks in there since the inception of the restoration project. And when we look at kind of a longer view of the harvest in Shinnecock, we see that in 2018 and 19, we were at levels harvested from Shinnecock that we haven't seen since 1987, or over 30 years. So has this increase impacted Shinnecock Bay? Well, if we look at brown tide in Shinnecock Bay, notice that there's been this decrease. So here's western, mid, and eastern portion of western Shinnecock. So here's kind of all of western and eastern Shinnecock. Looking at the far western area, we've seen this decrease blurb in 2016, but notice in 2018 and 19 and 2020, there were no detectable levels of brown tide. When we consider the closures to harvest, all of western Shinnecock was closed at some time in 2011, 14, and 15. In 17 and 18, only half of western Shinnecock was closed, so a reduction in half of the acreage closed. And in 2019 and 2020, there was no closure to shell fishing in western Shinnecock. When we look at the county data, kind of a historical record looking at light transparency and chlorophyll. The gray bars here are kind of the decade before restoration. And then since the 2012, we see that there's been kind of this deepening of the Seki in the middle and eastern portions of western Shinnecock. When we look at chlorophyll, we see that there's been kind of this direct, reduction in chlorophyll in all three areas of western Shinnecock. We created a biooptical model in 2017, looking at the amount of light reaching the bottom. The really important month is the month of August, when our temperatures are the warmest. And so this is the critical time for the plant to be able to get enough carbon to survive. And when we look at August, comparing 2017 to 2019, what we see is that there's been this change in light in these two areas, which correspond to where we have our spawner sanctuaries. And if you were to turn those into bathtubs and calculate how long it would take to filter that water through the hard clams that we've put in those areas, we'd see that it falls underneath that threshold by DAME of below 4-day turnover to have a biotic control of the water column. I think more visually effective, these are baited remote underwater videos. These are some sets that were done at the same spots in western Shinnecock, comparing 2014 to 2019. And if you could look through a series of these brews, they become clearer. Those brews in western Shinnecock are visually clearer in 2019 than any of them were in 2020. Again, kind of visually demonstrating the change in the light characteristics. But what impact has this had on seagrass restoration? Well, our restoration is we go out and we collect reproductive shoots in eastern Shinnecock Bay. We then separate those reproductive shoots, and we've put them into spat bags, and then we've attached those to buoys using the kind of the buoy deployed reproductive shoot method. Here you can see them being placed over in western Shinnecock. We then come back the following year, and we can see the grass that has grown underneath the buoys over there. So some of those areas are seed limited, and we see that by these areas underneath the buoys. But we've also have a strategy where we are now collecting and keeping the seeds until they fall from the reproductive shoots, and then deploying them out in October after the temperature has dropped and the seed predators are not consuming as many seeds that we hand broadcast in those areas. Now, here's a Google Earth image. You can see a number of areas where we've put our buoys and our seeds. Here's some seagrass, some eelgrass over in 2013, a year after we started our restoration efforts. And here is 2019. And so you can see kind of this grass now extending into areas much further to the east. And if we delineate that grass, we see that there's been a net gain of about 97 acres. And if we use kind of the conservative estimate from UNH or URI about the costs, we're somewhere around $24 million, what it would cost to replace that grass, restore that grass. Now, this caught the attention of our governor, and he has now established 5 other locations around Long Island where they are using a similar strategy to affect water quality by using hard clam spawner sanctuaries in these areas. So the highlights of this project, we have a continued hard clam population recovery in Shinnecock, the largest hard clam landings in 33 years, over a 750% increase since the restoration activities began. The highest density of adult and juvenile hard clams are documented by the benthic survey since the inception of the SHRP. Evidence supporting the hypothesis that the increase in clam population caused by SHRP restoration efforts, given the size structure. We have 5 consecutive years without dense brown tide, 3rd consecutive year without any brown tide. Seagrass has expanded over 97 acres in Western Shinnecock. This was driven by data, not politics, not by wishful thinking. We were able to create no-take zones for the spawner sanctuaries because of the ownership of the bottom by the town trustees. We used adult clams beyond a significant predation threshold. Seed clam mortality rates were incredibly high. The scale of the project matters, and continued long-term effort is necessary to see results. Thank you.